in the last class we looked at how protein structures are determined by NMR, and we also saw that we do not show one particular structure as a representative structure in NMR. We show what is known as a bundle or ensemble of NMR structures. This is shown here because all the distances and angle constraints that we gave would have been satisfied by all the 20-25 structures shown here. So, therefore, all of them are equally correct, and actually if you look at it, they even have all of them superimpose on each other pretty well, except this part of this protein here looks very superimposable, but that is because in this region there was not many constraints given to us. So, the software program could not determine a unique structure for this part. So, these are typically the loop regions, and the end of the protein typically they behave like this in NMR structures because they are not well defined, meaning there is no particular distance value we are able to find between atoms in this part because they are very flexible. So, the nosy correlations in the nosy spectrum are not very strong, they are weak. So, when the correlations are not present, so there is no distance information between neighboring atoms or far away atoms. So, therefore, this part of the protein is not having a well defined conformation, which is not bad, which is okay, consider is fine, because that shows it is a flexible part of the protein. Similarly, if you look at this helical part here, it seems to be well defined, and all of them are almost superimposable because the distance values are satisfied equally in all the 20-25 structures which are shown here. So, typically this is how NMR structures are published and shown, and we normally take the average structure or we can take the first out of the 25. Remember, the 25 structures here are given some ranking from 1 to 25, and the first best structure can be shown as the representative model. Okay. So, now the next part in NMR structure determination is to verify. So, remember we have solved the structure, but how do we trust that the structure is correct or not? How do we believe? How do we validate? So, therefore, this is a very important step in protein structure validation, where we have to see whether the structure is correct or not. So, typically this is done by many software. Normally it is done by the database where we deposit the structure. So, database already checks, does lot of pre-checking before it accepts your structure. If the structure is not satisfactory or violating some basic rules, then it is not allowed to deposit. So, what are the checks we do? Typically you check the bond lengths, bond angles, because a CH or a CC bond angle, a bond length should not vary much even if you have alpha helix or beta sheet. The CC, these angles and bond lengths are normally fixed whether it is any structure you take. So, one should check whether your structure which you have got at least is having the correct values for these angles and bond lengths. The next check is done is whether any NOE which you have given was violated or not. Violated means suppose you gave a distance value of 3.1 for 2 atoms, but then in the real final structure it was found to be 4.1, which is a huge violation. So, that is not acceptable. So, that kind of distances checks are carried out, and then you reject those structures where the violation or deviation between the given NOE distance and the observed distance is very high. So, there is some sort of a cutoff which is used, and without anything beyond that cutoff is rejected. Then you check the backbone angle phi and psi which falls in the allowed region. So, this is a very important plot which is named after an Indian scientist who worked in Indian Institute of Science, and he discovered this when he was in University of Madras, and this is a very important plot called Ramachandran plot. That is something I will discuss next. And the last thing is you have to check if the atoms are not coming too close to each other. So, this is called bad contact. So, remember two atoms, let me show in a diagrammatic way. Two atoms cannot bump into each other, this is not allowed. They have to have sufficient distance because this will cause a repulsion, and therefore, it is not in nature you can't expect two atoms to be closer than certain distance. So, that is called a bad contact, and if this happens then your structure has to be rejected, it has to be. So, again for all this there is a cutoff, means it is not that it is yes or no, there is a cutoff. So, threshold. So, if it is beyond a threshold then it is rejected, but if it is within a threshold it is acceptable. So, let us look at now at this important plot called Ramachandran plot. This is basically what was invented. The idea was invented by Professor G. N. Ramachandran, and the idea is follows that look at this here on the right hand side is drawn the backbone structure of a amino acid of a protein chain. So, this is something we have already seen, and you see there are what is called the torsion angles. So, this is a torsion angle phi between the HN H alpha, and this is a torsion angle psi. So, these phi and psi are very important indicators of secondary structure. So, it turns out he did lot of calculation on different structures, and he found out that if you are this chain if is in alpha helical geometry conformation, then the phi and psi value should fall in this region only. So, for example, let us say this is some amino acid lysine, and if the lysine is in alpha helix, then that phi and psi of that lysine should be in this zone, yellow and red color zone. Okay. And if it is in a beta sheet conformation, then it has to be in this only, and if it is a left handed alpha helix, which is normally rare, but it is still possible, then it will be here. So, you see that this phi and psi are very highly restricted, they cannot take any arbitrary values, they have to fall in this zones of secondary structures only. But the only amino acid which is exempted from this criteria or it can have any value is glycine. So, glycine can come anywhere in this picture, but other than glycine all other amino acids should fall in. And why is glycine exempted? Because glycine is a flexible amino acid, it has only 2 protons, remember here there is no C beta. So, this restrictions what we are seeing here is actually coming because of the steric factor. Steric meaning there is a restriction because this C beta and N and all these atoms cannot come very close to each other. So, because of these geometrical restrictions there is a restriction on the phi and psi, and that is reflected in this picture. So, that was a very important plot, and now in every paper, every protein structure which we publish, one has to show this graph and tell that look, for my protein which I have solved, all the phi psi value are coming in the right place. So, if I claim that my protein is a beta sheet protein, I will show that everything is here. If I claim it is a mixture of alpha and beta, then alpha part should come in this, and all amino acids for the beta part should come in this side. So, this is called structure validation. You are validating with this plot whether your structure what you claim is correct or not. For example, if you claim it is a beta sheet, but then if the phi and psi of your beta sheet is coming somewhere outside this, then it is getting violated. So, that is what are the checks which are carried out. So, these are all carried out by software which are available in the database. So, this is the database which I mentioned earlier. In this database all the structures are deposited, and here itself the checks are carried out before you deposit and before it is released for the public. So, this is basically brings us to the end of the secondary structure part, I mean sorry, the structure determination by NOE or by the distance information wise. But there is another method which I told you in the beginning, is that we do not have to go through this laborious procedure of NOE. Remember, NOE distance determination or sorry, structure determination is quite laborious. It takes lot of time because the 3D NOESY experiment is not something very easy to interpret, number 1. Number 2, as I said, it is an iterative procedure. You have to give distance information, get to a structure, find out what are the violations, why the distances are violated, what could be going wrong, and then go back, start again and look at a different set of NOEs or remove some bad NOEs, and so on so forth. So, that takes quite a long time if you want a very good structure. But if you look at computational method, these are newly emerging methods which have come up in last 10 years or so. Here what you do is you first you assign the protein. So, assignment is something which I said is mandatory, you can't avoid that step. So, once you assign a protein, then you have the protein chemical shifts with you, take that chemical shifts and combine with homology modeling, meaning the sequence which you are trying, the structure which you are trying to solve, a similar structure may already be available in the database, which database PDB the database. So, if the similar structure, similar sequence, a structure of a similar sequence protein is available, then you do not have to really solve that structure again, you can use homology modeling, but the information you are going to give extra is the NMR chemical shift of your protein which you are trying to solve. So, this is an important point here that one should look at the homology based modeling if the structure is already known for a similar protein. So, there are different software, we will not go in this course particular course on all the details of this, because this is quite involved in terms of the algorithms methodology that is used. So, we will just I want to name two softwares which are popularly used. One is called CS Rosetta, which combines NMR chemical shifts with homology modeling to get the 3D structure. So, you can look at this website where you can download and use it for your protein, or upload your data and they will solve the structure. Similarly, CS2 to 3D, CS2 means chemical shift to 3D structure, combines NMR chemical shift with structure prediction tools. Okay. So, there are many structure prediction tools in bioinformatics which are known and well established today. Those can be used and combined with NMR. So, essentially what we are doing here and also here is we are using computational methods. In this case it is homology modeling, but in this case it is ab initio, or it could be a structure prediction method. But what we are doing is, once we get a structure or when we are predicting the structure, we are constraining it, means we are trying to constrain such that the chemical shift what we have observed should be satisfied. Okay. So, each time when a structure is predicted or a homology model structure is generated, a chemical shift is predicted from the structure, and what the chemical shift prediction and the observed chemical shift should match. So, that is a kind of a constraint which is given to this software and they generate the structure. So, this is another alternative to NOE, but again the resolution which we get from these may not match the one which we get from NOE. So, NOE is still the more preferred way, but that is a slow approach, this is much faster approach. Okay. So, we come to the next part of this course where we look at now, having assigned a protein, having known the structure of the protein, can we do now study, can we do something to study protein to ligand interaction. So, this is a very important topic because most of the drug discovery depends on this interaction. So, we are trying to suppose discover a new drug molecule, we need to know if it binds the target protein or not. So, these are the different techniques which we will try to see in this course, and to know what is the ways, different methods which we can use to characterize. So, our goal here is to characterize or figure out whether a particular ligand is binding to a protein or not. Okay. So, let us start from the first most basic experiment or most basic method called chemical shift perturbation. So, we have already seen this chemical shift perturbation in the previous part of this course, where I showed you for HSQC how HSQC is useful for perturbation for ligand interaction. So, I am repeating a slight repetition of that. So, typically chemical shift perturbation means we are trying to find out what chemical shifts are changing in an HSQC spectrum. Again specifically we use N15 proton HSQC. So, N15 proton HSQC or HMQC can be used, and then we titrate means we add the ligand and then try to monitor the changes happening to the protein signals. So, what this HSQC is now for the protein, which means I should know, I should label the protein with N15, number 1. Second, I should know the chemical shift of every peak in that protein. So, again this brings us back to the importance of assignment. So, suppose let us say this is a protein and it is interacting with a drug molecule or any ligand, it could be another protein, a peptide, any other partner, binding partner. So, let us say they interact with each other and it forms a complex. So, what we do is we study this complex formation by HSQC, that is N15 HSQC or HMQC. So, in HSQC or HMQC, if you recall, by now you should be now familiar with this, that we observe the NH correlations in the spectrum. Okay. So, these NH correlations are seen. Now, if this for a given in the protein chain, let us say this is a big protein chain and the amino acids which are on this interface, means sitting on the binding interface with the ligand, they are expected to change chemical shift of those amino acids, these amino acids here and here are expected to change when the ligand binds to the protein. Why do we expect it to change? The reason is that there is a small change in the structure will happen at the interface. Not only that, the chemical environment before adding the ligand of these amino acids here will be different from the chemical environment when the ligand is bound to the protein. So, therefore, there is a change in the chemical environment or change in the structure can lead to changes in this backbone NH chemical shift. Of course, all chemical shift will change, but we are only looking in HSQC spectrum because it is a very convenient, very easy to do the experiment, and in that we see each NH correlation they are expected to shift meaning change. We will show these examples soon, we will see some real case studies, but those shifts are what is indicating whether the amino acid if the ligand is binding or not. Okay. So, let us look at this in more detail now. So, there are different types of ligand binding. So, this is basically the amino acid residues. So, let me go back to this here. So, as I mentioned with these arrow marks here, they are pointing to the interface. So, let me write this word very clearly. This is called a binding interface. Okay. So, it is a binding interface, and the amino acids which are located in this interface now will experience a difference in the chemical shift before and after the ligand interaction. So, this change in this chemical shift is what we are looking for. Okay. Now, what about the amino acids here inside the protein? There we do not expect much change because it will not be affected by a ligand which is sitting far away. Only the one which are close to the interface, binding interface, these are the one which we expect to really undergo a big change in or some change in the chemical shift. Okay. That change will depend on what kind of ligand is this. Okay. It will depend on how much structural change is happening and so on. It is not that every time you will get the same type of changes for every protein. It depends from protein to protein, it depends on ligand also. Okay. So, the amino acids which show significant changes in your spectrum will be the one which are assumed to be at the site of ligand binding. What is the site of ligand binding? The site of ligand binding means binding interface. So, binding in the interface, the residues which are located on the interface of the binding will be the one which will show significant changes. Okay. So, let us take an example. This is a real case example of a protein. So, you can see these are all this is HSQC spectrum N15 proton, and this red color is a bound protein and the blue color is coming from the free protein. So, free means the normal protein, and after adding the ligand the peaks have moved. You can see here, if you zoom this dotted box here, you can see in this here there are changes, shifts happening. Shift means they are all moving. So, the blue has moved here, this blue has moved there and so on. So, these are the changes happening in the HSQC spectrum of this protein when the ligand was added. Now, you can see here these numbers E151 and so on, these are the residue numbers, they are the ones which undergo change. That means, I should know which peak here corresponds to which residue, and that I would expect it to be knowing at this stage because I would have assigned the protein. So, I will be knowing all the assignments of each peak. So, now this changes what you are seeing here can be plotted in a graphical manner. So, what you see here in this graph on the right side is that there is a residue number on the x axis, and this vertical blue color bars are telling me how much chemical shift change is happening. So, I can write it as delta delta. Delta delta means how much change in the chemical shift is taking place. So, this change is what you see here. But if you notice, the changes here is happening in both N15 direction and the amide horizontal direction. So, that means the change is not just in the amide region, I mean amide chemical shift, it is also change in the N15 chemical shift. So, how do we combine this change in the two dimensions? We combine with some formula. So, this is shown here. So, for example, this change delta delta. So, this is got cut here delta delta in ppm scale can be calculated like this. So, you have a chemical shift of proton in the bound form minus chemical shift of the proton in the free form for a given amino acid. Take for example, R156, you calculate this difference for that amino acid between the red and blue color, the proton dimension, proton axis means the horizontal, this horizontal axis. Then you add the shift in the nitrogen axis. Nitrogen axis is N15, the small shift here you see if there is a vertical shift also for between this R56 red and blue, and then that difference in the N15 dimension is also taken, but it is scaled down because of the gyromagnetic ratio of N is small. So, the scaling down is done and then added to this, and then use the square root, you calculate the final delta delta, and that is what is plotted here in this graph. So, you can see for the residues between 150 to 160, this part, that is this portion, seems to be changing a lot. Okay, it is beyond the threshold, threshold meaning the standard deviation. So, therefore, I can say that this is my binding interface. So, I can say this is my binding interface, because the binding interface is what is expected to show a significant shift when you add the protein, the ligand to the protein. So, this is basically what is, this is how we do normally all the chemical shift perturbation analysis. You add the ligand to the protein, first you record the spectrum, the HSQC spectrum of the protein alone, N15 labeled, and once you have assigned the protein, once you add the ligand to that sample, and then the sample, the ligand will now cause a change in the chemical shift if it is binding, and wherever the changes are taking place, that can be plotted graphically, and wherever there is a cluster, means a group of changes, that group is involved in the ligand interface binding interface. Okay. So, but now let us see further, there are some little complications which can happen. First of all, one should know what are the different types of interaction. So, a protein to ligand or a drug molecule, there are possible interactions are, it could be electrostatic in nature or driven by electrostatic interaction, or this interaction could be driven or dominated by hydrophobic interactions. These are the two broad possibilities. Okay, so, how do we know it is electrostatic or not? We can find out like this. Suppose the binding interface on the protein could be a positively charged, residues or surface could be positively charged, and this side of the ligand could be negatively charged. So, when these two interact with each other, the positive surface of the protein interacts with the negatively charged surface of the ligand, and that means it is an electrostatic interaction. So, that can be found out by based on the charge of the surface where the protein, the ligand is binding, and based on the charge of the ligand. So, that is possible, and that can be also the opposite. I can have this as negatively charged or this, and this could be positively charged. So, it could be opposite, but still the interaction is driven, means governed by the electrostatic interaction. So, this is I will give you a real example from a publication. You can go to this paper to get details, but what was seen here is this is a protein called ubiquitin. So, when you add a silver nanoparticles, silver nanoparticle is a negatively charged system, it goes and interacts with the positively charged surface. You see the blue color here is a positively charged surface of the protein, and these amino acids which are indicated are the ones which show very high chemical shift perturbation, delta delta, that is what we saw in the previous slide. So, those which show undergo very high delta delta, they were plotted on the surface of the protein, and it was found that this all of them lie, means are located on the positive charge surface, blue color is positive, of the protein, which means this nanoparticle is interacting, because it is negative, it is interacting with the positive surface of the protein, which means it is this interaction between the nanoparticle and the protein is driven by electrostatic interactions. Okay. So, this is very important to know. This is how NMR can give information on what is the nature of interaction between two ligand and a protein. Based on the chemical shift perturbation plot, one can figure out from the structure where it corresponds to, whether it is on the positive side or negative side, or it could be hydrophobic amino acids, and in such cases it could be hydrophobic interactions. So, that kind of information comes out from such analysis like this. Okay. Now, the next question you may want to ask is, okay, I know there is an interaction, but is it a strong interaction or weak interaction? How do I find out that the ligand is binding strongly or weakly? So, this is shown in a quotes, because strong and weak are always relative to some measurement. It is not a absolutely strong or absolutely weak, relative to something, but how do I still figure out, or how do I characterize whether a ligand is binding weakly or strongly to a protein, and that can be answered based on the concept of chemical exchange. Chemical exchange is a very important concept in protein NMR. We will take it up in the next class, where we will see how this helps us to find out whether a protein and ligand are having a strong interaction or weak interaction. So, we will see that in the next class.